bioengineering. I decided to make you guys a video about ELISA because I've been trying to find a really good video on YouTube that was simple enough as an introduction, but had enough detail that you would understand the biochemistry and the molecular biology that was happening. I couldn't really find a video that I thought really captured both of those two criteria, so I decided to make you a video explaining how ELISA works, and we're going to focus on the experimental design because a lot of science is trying to see something that you can't see, and you have to make it so that you can see it. And that's exactly what's happening with the ELISA assay as a test for coronavirus. We talked about that in class, and we also discussed a little bit why it takes a while to get ELISA tests to work properly. So just sit back and I'm going to draw you a couple pictures because if we were on campus, I'd be able to draw it on the board and you would understand it very, very well. And plus I would give you some chocolate, and that always makes science a little bit easier to understand. Welcome back, bioengineering. I am going to draw a couple pictures for you to explain how the ELISA assay works. So ELISA is a technique that uses molecules from the immune system to detect another molecule. So for the coronavirus, this is going to detect in a patient's blood if they have antibodies for the coronavirus in their bloodstream. And the only way you would have that is if you had been exposed to coronavirus. And the way you perform the test, I showed you in class a picture of the microtiter plate that has all the wells. So if we're looking from a side view, then we have a plate that has lots and lots of wells. And part one for ELISA, you add some of the virus that you are trying to detect. So I'm going to make that into some triangles. Okay, so you add first pieces of the virus. So that would be some of the DNA, some of the RNA, some of the proteins, etc. Anything that would alert your immune system that something was there that was not supposed to be there. And a general term for any kind of molecule that would send a signal to your immune system that it's foreign, those are called antigens. All right, so that's just a generic term. So if you've got the common cold, then the parts of the common cold inside of you that are triggering your immune system, those are called antigens. So anything that's not supposed to be there is called an antigen. All right, so the first thing, we put in some of the antigen that we're trying to detect. And we're not going to detect the antigen directly. We're going to be detecting the antigen indirectly. So for our lab, you're going to learn indirect ELISA. There are other forms of ELISA, different ways to do an ELISA experiment. There's direct ELISA and sandwich ELISA. So we're going to actually be learning the indirect ELISA. Now, after we add our antigen, so if we were trying to detect HIV, we would add little HIV antigens or West Nile virus antigens, etc.
And the antigens are going to stick to the wells. Then we rinse out, we rinse out and dump out the liquid that we had in our wells. And now all that's in the wells are the antigens. There's no liquid in here anymore. And the antigens have completely coated the well. The next step is then add the patient's sample. Okay, so when we add the patient sample, the antigens from coronavirus or whatever we were studying or wanted to detect, that's still stuck on the side of the wells. And when we add the patient sample, their bloodstream is going to have antibodies to other foreign viruses, bacteria, etc., anything that was not supposed to be there. They have those in their bloodstream, okay, because that's their job, is to detect anything that's not supposed to be there, and then that alerts the rest of the immune system that they need to attack and destroy. So if the patient has been exposed to coronavirus, so if this patient has been exposed and when they were exposed, they made this brown antibody. Okay, so we're going to say that the brown antibody is the antibody for the virus that causes coronavirus, or COVID-19, sorry. Okay, so they would have the brown antibody in their bloodstream. You let it sit for a little bit and give time for any of the antibodies that can stick to the antigen have time to stick to the antigen. So what happens is the brown antibodies or the coronavirus antibodies are going to stick to the antigens, but the antibodies for other foreign substances do not stick. When we rinse the wells, we dump out the liquid, we rinse with some buffer, and these are called washes. And the goal is the washes will wash everything out except for whatever is stuck to the sides of the wells. We know that the antigen is always there. We put that there first on purpose. What we don't know is if any antibodies for coronavirus have also stuck to the sides of the wells by sticking to the antigens. We can't see that with the naked eye, but we're going to add something that we can see. And what we're going to add is called a secondary antibody. And this secondary antibody binds to the coronavirus antibody. So let's pick a new color. We'll do orange. So we have what we call a secondary antibody, and it recognizes the coronavirus antibody. It will stick to the coronavirus antibody. And this secondary antibody has something very special attached to it, which we'll talk about in just a second. So our patient sample has been added to the plate. And this patient has been exposed to coronavirus, so their antibody sticks to the coronavirus antigen. We've washed the plate out with buffer, so the only thing that can be in the wells right now is anything that's stuck to the wells. And the only way anything can stick in these wells is if they are attracted to each other. It's very, very specific. When we add the secondary antibody, all right, so we pour in the secondary antibody, and the secondary antibody does not bind to the antigen, but it does bind to our coronavirus antibody. So remember, it's got that special piece that we haven't talked about yet. Now, we let the secondary antibody it's called incubate. We let it sit for about 5 minutes, and that gives the secondary antibody time to bind to any coronavirus antibody that happens to be there. So our patient was exposed to the coronavirus, so they do have the coronavirus antibody. The secondary antibody only binds to the coronavirus antibody. So now when we do our wash, when we dump out the liquid in our microtiter plate, the only thing that can be left in the well after we dump out the contents and we wash with buffer, we know that the antigen will be there. We know that that sticks. We still don't know if there's any coronavirus antibody on there, but we do know if there is coronavirus antibody here, the secondary antibody will also be there because it sticks to the coronavirus antibody. Now, that molecule that stuck to our secondary antibody, this right here, this is an enzyme. So this molecule is an enzyme. Remember, we can't see any of this still. We still can't see this. It's all there, but we can't see it. The enzyme is what's going to allow us to now see what's happening in here. This enzyme is chosen because the reaction that it catalyzes causes a change in color. So while we've been adding all of our samples, we've been washing everything out. At the very last step, we're going to add the substrate for the enzyme. And you learned in biology that substrates, when an enzyme is present, it increases the rate of reaction for that reaction. And what is produced are products. And what's special about this enzyme is the fact that the substrate for this enzyme is a different color than the products. So let's say the substrate was green. And the products, let's say, turn purple. When we add the substrate, I'm just going to draw one antigen this time. When we add the substrate for the enzyme, if the patient had coronavirus, then they would have the antibody for coronavirus. And that antibody would stick to our antigen. If our patient had coronavirus, they have the antibody for coronavirus, and our secondary antibody sticks to the coronavirus antibody. So if the antibody for coronavirus is present, then after all of the washes, our secondary antibody, which has the enzyme, is also present. I need to make my wells a little bit bigger. Okay, we've done our washes. The only thing that could be in the wells now are things that are stuck there, all right? Now, when we add the substrate for the enzyme, the sample is green, okay, because the substrate's green. It will stay green unless the enzyme's there. If the enzyme is there, the substrate reacts with the enzyme like it normally does. And as it reacts with the enzyme, it will change color. It will change from a green to a purple. So as you let the sample sit for 5 minutes or so, if it changes now to a purple color, then the enzyme had to be there, because our substrate does not change color without the enzyme. And the only way the enzyme can be there is if the secondary antibody's there, because the enzyme is stuck onto the secondary antibody. So that means if our sample changes from green to purple, the secondary antibody also had to be there. But the secondary antibody can only be there if it's stuck to the coronavirus antibody. And the only way the coronavirus antibody can be there is if it was in our patient sample here. So if our sample changes from green to purple, we know that the patient has the antibody in their bloodstream. If the sample would have stayed green, that means our patient has not been exposed to coronavirus. Because if you don't have that original coronavirus antibody bind in the first step to the antigen, if it's not there, then the coronavirus antibody doesn't bind. If there's no coronavirus antibody, then the secondary antibody has nothing to stick to. So it just gets washed away. And it has the enzyme on it. So when we add the substrate doesn't even see any enzyme at all, because the secondary antibody is not there. The secondary antibody is not there because there was nothing for it to bind to. The patient had never been exposed to coronavirus. They didn't have the antibody for coronavirus. And the secondary antibody only sticks to the coronavirus antibody. It is a very interesting experimental design. It's a great example of how do you see something that you can't see. And you see the rationale here of this cascade of criteria that have to be met to get this color change. Now, what takes a long time to develop an ELISA assay, first is to get exactly the right piece of the antigen that the immune system recognizes. There can be some variability here. And another place that's a holdup is this secondary antibody. You will have to grow this antibody probably in some animal, and then attaching the enzyme onto this antibody can change the shape of this antibody so that it doesn't recognize the original antibody anymore. So there's quite a bit of work here in producing the secondary antibody and then getting the enzyme on that antibody. So those are probably the two, those are two of the main sources that can cause the development of these assays to take longer than expected. All right. If you have any questions about this, let me know. When you answer questions about a positive or negative result in ELISA, you'll need to go through this cascade of reactions that occur in detail. You're going to feel like you're stating the obvious, but please state the obvious. Describe it just the way I have. If there is no color change, that is because the substrate for the enzyme could not be converted into the products because the enzyme wasn't there. And if the enzyme's not there, the secondary antibody's not there. If the secondary antibody's not there, then the antibody for coronavirus is not there. And if the antibody for the coronavirus is not there, that means the patient didn't have the antibody to the coronavirus in their sample. So you want to describe the results in a way that shows that you understand what causes a positive result and what causes a negative result. So explain it more than you think you need to. If you feel like you're stating the obvious, you're doing a good job. And if you have any questions, just let me know.